This video tutorial is all about biotechnology, and in this tutorial we're going to look at the use of microorganisms in biotechnological processes. Firstly, we're going to look at the scaling up of drug production. Commercial drug production uses large stainless steel containers called fermenters, in which growing conditions can be controlled to ensure the best possible yield of the product. Now, the conditions that need to be controlled include temperature. You don't want it to be too hot or the enzymes will denature. You don't want it to be too cold or growth will be limited. Nutrient availability. So you need those microbes to have supply of carbon, nitrogen, minerals, vitamins, so that they can grow and synthesize the product you want. You need good oxygen availability, because most of the microorganisms you want to respire aerobically. The pH needs to be tightly controlled for sort of optimum enzyme activity, and hence the growth and synthesis of the product you're making. And the concentration of the product itself needs to be controlled. So if the product is allowed to build up too much, that might actually affect the synthesis process. Now, a fermenter must first be sterilized using superheated steam. It can then be filled with all the components required for growth and supplied with a starter culture of the microorganism to be used. The culture will then be left to grow and synthesize the products. There are two different types of culture. There's batch culture and continuous culture. First, we're going to look at continuous culture. Some products are synthesized by the microorganism during normal metabolism, when they are actively growing. These are called primary metabolites. Such products are continually released from the cells and can be extracted continuously from the fermenting broth. The broth is topped up with nutrients, as these are used by the microorganisms. Some of the broth is removed regularly to extract the product and remove cells from the broth. Otherwise, the population becomes too dense. So this is known as continuous culture and keeps the microorganism growing at a specific growth rate. Now we're going to think about batch production, batch culture. Other products are produced only when the cells are placed under stress, such as high population density or limited nutrient availability. These are called secondary metabolites and are produced mostly during the stationary phase of growth. We'll look at that a bit more in a different video tutorial. Hence, the culture is set up with a limited quantity of nutrients and allowed to ferment for a specific time. After this time, the fermenter is emptied and the product can be extracted from the culture. So that is batch culture. Asepsis is ensuring that sterile conditions are maintained. The nutrient medium would also support the growth of unwanted microorganisms, which would reduce production, because the unwanted microorganisms would compete with the cultured microorganisms for nutrients and space. They would reduce the yield of useful products. They'd spoil the product you're making. They might even produce toxic chemicals or destroy the cultured microorganisms and their products. In processes where food or medical chemicals are produced, all products must be discarded if contamination by unwanted organisms occurs. Flory and Chain devised the process to successfully mass-produce penicillin through fermentation by the fungus penicillin. Modern strains of the fungus have been selectively bred to be more productive than the earlier strains. Penicillin is a secondary metabolite. It is only produced when the population has reached a certain size. Therefore, penicillin is manufactured by batch culture. So the fermenter is run for sort of 6 to 8 days. The culture is then filtered to remove the cells. The antibiotic is precipitated as crystals by the addition of potassium compounds. The antibiotic may be modified by the action of other microorganisms or by chemical means. The antibiotic is mixed with inert substances and prepared for administration in tablet form, as a syrup, or in a sort of form suitable for injection. Citric acid, or citrate, is widely used as a flavour enhancer, a preservative in manufactured foods, and an antioxidant. It is produced as an intermediate of the Krebs cycle under aerobic conditions. Citric acid is mass-produced by continuous fermenter systems from cultures of the fungus A. niger. Carbohydrates are continuously introduced into the fermenter in order to maintain this citric acid production. Iron is excluded from the mixture in order to slow the further conversion of citric acid within the Krebs cycle. As the citric acid accumulates, it is extracted as part of the medium that is continuously being withdrawn from the fermenter. Insulin is widely used to treat type 1 diabetes. It was previously extracted from the pancreas of animals such as cattle or pigs as they were sent for slaughter. Insulin from slaughtered animals is not identical to human insulin, and so it's less effective than human insulin and is also very expensive to extract. In 1978, synthetic human insulin was developed by genetically modifying a bacterium. The gene for human insulin was combined with a plasmid to act as a vector, so the gene could be inserted into the bacterium E. coli. The resultant genetically modified bacterium enabled the production of vast quantities of human insulin at relatively low cost. Insulin is manufactured by continuous culture. Finally, we're going to look at bioremediation. Bioremediation is the use of microorganisms to clean the soil and underground water on polluted sites. The microorganisms convert the toxic pollutants to less harmful substances. The idea started when a bacteria was modified back in 1971, enabling it to break down crude oil. And it's proposed that this bacterium could be used to treat oil spills. Solvents and pesticides can also be treated using bioremediation. Bioremediation involves stimulating the growth of suitable microbes that use the contaminants as sources of food. Now, it requires the right conditions for the growth of the microorganisms. So you need available water, suitable temperature, suitable pH. And when these conditions are not suitable, they need to be modified through the addition of suitable substances. So in some cases, additional nutrients such as molasses may be needed to ensure the microorganisms can grow effectively. Or it might be necessary to pump in oxygen for aerobic bacteria. Where conditions cannot be made suitable in situ, the soil can be dug up and moved to be treated ex situ. Now, there are lots of advantages of this process. It uses natural systems. It's not very labour intensive or equipment intensive. The treatment can usually happen in situ, doesn't have many waste products, and is less risky in terms of fewer sort of personnel are exposed in sort of cleanup jobs. However, bioremediation is only suitable for certain products. So heavy metals cannot be treated in this way.